Hello and welcome back to educator.com, and remember this is our molecular biology course. So today we're going to talk about the structure of proteins. As an overview, we have to first talk about amino acids since those are the monomers of proteins. We'll talk about the types of bonds in proteins called peptide bonds. And then importantly, we'll talk about the big types of organization, of structure. We'll mention protein folding and briefly talk about post-translational modifications. Finally, we'll end with protein classification and a short introduction on enzymes being biological catalysts, just as we mentioned in the first lecture. So amino acids, as I said, are made up of the monomer, or are the monomers that make up proteins. And this is the general structure of an amino acid right here on the left. And as we see here, we have a carboxylic acid group, we have a carbon bound to that, which is our alpha carbon. Okay. Then bound to the alpha carbon is our amino group. All right. Now, the carbon, its third bond, the alpha carbon, it makes with a hydrogen. And finally, its fourth bond is to something else. All right, so what we call an R group or a side chain. Now, the amino group, the hydrogen, and the carboxylic acid group all attached to the alpha carbon is going to look the same in all of our amino acids. The only difference is going to be in the R group. Now, importantly, the acid dissociation constant of the amino group is at 9.1, and the acid dissociation constant of the carboxylic acid group is 2.3. This will come into play a little later. Now, amino acids are going to make up proteins. They do this by making peptide bonds, also known as amide bonds. And the addition of amino acid 2 to amino acid 1 is catalyzed, and the type of reaction is called a condensation reaction, right? In which case we are pushing out water. So this right here is your water molecule, that gets pushed out. And as we can see here, the N-terminal, okay, or the N portion of amino acid 2 always comes in and makes its attachment, its bond, to the C-terminal portion of amino acid 1. So that's why we have amino acids following an N to C-terminal orientation. So a protein is N to C. It's basically like saying N is the N-terminal amino acid is the first amino acid that's put in, and the C-terminal amino acid is the last one that's put in. Here's a table of the 21 amino acids that we find in proteins. Now, many of you may have learned that there are 20 amino acids found in proteins, and that is very correct. The one that makes the 21st is selenocysteine. And that is just a cysteine molecule that has a selenium group instead of a sulfur group. All right. But this has also been found to be incorporated into proteins. So these are all of the 20 amino acids that we incorporate into proteins. All right. There are many other types of amino acids that are not incorporated into proteins. Now, our amino acids can be broken into multiple groups. We have our charged amino acids, all right? We have our positively charged or basic amino acids. We have our negatively charged or acidic amino acids. So only 5 of the 21 have any type of charge, right? Now, we have our uncharged amino acids right here in group B. We have in group D our hydrophobic side chains. All right, so these are the ones that are going to participate in our hydrophobic bonding. And then we have our special cases. So as I said before, selenocysteine is kind of a special case, and cysteine is very special in that its sulfhydryl group right here can participate in what are called disulfide bonds, and these are very important for the structure of proteins. Glycine is special in that it is the simplest amino acid to where its R group is only a hydrogen. And then proline is specific, is a special case in that it actually has a secondary amine. So its R group is not free. It comes back and binds with the amine group. And we'll know this later, being able to call this a helix breaker. So when we talk about our amino acids, we need to know that our ionization will vary based on the environment, the pH. Right? So we have our cation. All right? Now, a cation is just another way of saying a positively charged ion. An anion is a negatively charged ion. Okay? That's what we normally think of. Now, our cation group is when we have our amino group being protonated, so it has an extra charge right here. We have this extra positive charge, all right, because it has an extra hydrogen. And we still have the carboxyl group having its proton. Okay. Now, this can only happen when we are at a pH below the acid dissociation constant of that carboxylic acid group, okay, as well as below the amino group. Now, if we remember right, the pKa of this is 2.3, and the pKa of this is 9.1. So, when this is below 2.3, okay, so when the pH is below 2.3, we're in the cation form. All right? Now, when we increase our pH, okay, past that 2.3, we are going to start getting into what's called the Zwitter-ion form. And this is going to exist between the pKa of our carboxylic acid group and the pKa of our amino group. So this is when our pH is between 9.1 and 2.3. All right, so when the pH is like this, okay. All right, so this is when we're going to see a Zwitter ionic form. And what that means is that the carboxylic acid, if we see over here, it no longer has its H over here. So now it has a partial negative charge. All right, so we have a molecule right here that has a negative charge and a positive charge. And once again, this is when we're not talking about the R group at all playing a part into it. Okay, that is going to be completely separate. Now this is what we call a Zwitter ion, and a Zwitter ion is a molecule that has both a positive and a negative charge on it. Now this is going to exist until we have a pH above 9.1. So right here, when the pH is greater than 9.1, we're going to move into the anionic state. All right, and that's when our amino group loses this proton. Okay, right here, remember, here's that proton. We no longer have it. Okay, we no longer have it right here. All right, so we have our negative charge, Okay, we don't have that positive charge anymore, so this is a negatively charged ion. So a negatively charged ion we know as an anion. Now remember, we're not talking about the R group at this point. We're just talking about the major backbone. All right? So this is important. Now, what is the physiological pH in a human cell, in a human body? All right, it's about roughly 7.4. So we're always going to see, in physiological circumstances, we're always going to see our amino acids in the Zwitter-ion form. All right, so they'll have both a positive and a negative charge, meaning it's a neutral charge. And the only thing that will affect whether it's a positively or negatively charged amino acid is the R group. Good. Now, for our first example, if we can just do a little bit of memory recall, which amino acids would have a net charge at physiological pH, which, as I said before, is about 7.4? Well, what that's saying is what amino acids have a negatively or positively charged side chains, because that's the only thing that's going to be attributing to the charge, because the Zwitter ionic form is a neutral charge in and of itself. So our acidic ones, all right, otherwise known as negatively charged, which ones are they? All right, they are aspartate, all right, and by the way, the single letter code is D. And then we have glutamate. And its single letter code is E. All right, aspartate and glutamate are also often referred to as aspartic acid and glutamic acid. Okay, so now what about our basic amino acids, otherwise known as having a positive charge? We have 3 of those. All right, so we have arginine. Okay, that is R. We have histidine. Okay, and we have finally lysine, and that's a K. All right, so those are the only 5 amino acids that will have a net charge at physiological pH. Now, when we talk about amino acids, we need to talk about what form they're normally found in. Now, most of our amino acids occur as what are called L-alpha amino acids. So this is a type of configuration. Now, only L amino acids become incorporated into protein, all right? And we will see that the difference between an L and a D, or an alpha and a beta amino acid, actually look like. So what do they look like? All right, so here's going to be an example of an L or a D amino acid. And really all it is what side the amino group is on. Okay, so this is an L. And it also happens to be an alpha. L-alpha amino acid. And let's draw a D. Okay, so what we're looking at is there are plenty of other ways to see this, but very simply written out in planar form, if the NH2 is on the left, it's an L amino acid. If it's on the right, it's a D amino acid. Okay? All right, now what about the difference between alpha and beta amino acids? Well, let's do an example of a general example, and then we'll do a specific example. So for our alpha, okay. Okay. So what makes this alpha? You're looking at where this NH2 group is connected to. Now this NH2 group is connected to the carbon that is connected to the carboxylic acid group. So the way that we count, we would find our carboxylic acid group, and we find the carbon that's attached to it, and we call that carbon the alpha carbon. Okay? And therefore we have the NH2 group attached to that carbon, okay, being the alpha carbon. Now if we go to the beta, the beta amino acid, right? So we have the beta amino acid. Okay, so if we look once again, we count away from the carboxylic acid group. So this would be the alpha carbon. This would be the beta carbon. And this is the carbon where we have the NH2 group and the R group attached. So this makes it a beta amino acid. All right, so this is how we can tell between L and D, as well as between alpha and beta amino acids. Now, it's important that we know the side chains of all of our amino acids because they basically tell us what types of reactions these amino acids are going to take a part in. So the characteristics of our side chains, right, our R groups, if they're nonpolar, these groups are going to form or can form hydrophobic bonds, right? And these residues are often going to be buried in the interior of the proteins because they're hydrophobic. Now, in contrast, our polar groups, they can form hydrogen or ionic salt bridges, okay, or ionic bonds, which are called salt bridges. Okay, so they can form ionic or hydrogen bonds, and they often cluster on the surface of a protein, okay, because these are hydrophilic. So if, for example, we have a protein, and we'll make it easy, and we'll make it look like this, okay? You might be more likely to see our nonpolar residues on the inside and see our polar residues on the outsides, okay, on the outsides. Right? Now, if we get a little more specific, we can look at the hydroxyl groups on our serine, threonine, and tyrosine amino acids. They can be phosphorylated, meaning they can get a phosphate group attached. Now this makes them negatively charged, and it can also alter their function, either in a positive or negative way. Now the sulfhydryl group on cysteine is unique in that it can be oxidized to form a disulfide bond. So a disulfide bond is just a bond in between two sulfur groups, and this is going to be important for our structure of our protein. Right? Now we can have covalent bonds to carbohydrates, forming what are called glycoproteins, and they can be either O-glycosylated or N-glycosylated proteins. And all that means is the sugar making a bond to the amino acid via an oxygen or a nitrogen. And what we have here is that we can see these groups, this oxygen-hydrogen, right, which we know is a hydroxyl group, from serine and threonine can participate in the O-glycosylation. And then the NH2 group, the amine group of asparagine, can also participate in glycosylation. But in this case, it's an N-glycosylation. And finally, we have proline having what's called a pyrrolidine structure. And that is this big hunk of a secondary amine. Okay, so it has a nice, or not very nice, okay, structure in the fact that it's not an open chain. Now this is what we call a helix breaker. Prolines are not found in helices because this is too bulky, it's too big to be formed properly in a nice helix. So often they are found at turns, all right, because they help with a U-turn type characteristic. Now, our peptide bonds are also called amide bonds, as we stated earlier. And remember, they come from a condensation reaction where water leaves. All right, so the incoming amino acid, 2, makes a bond using its nitrogen, right? So it's a amine group with the already existing amino acid 1's carboxylic acid group. All right? And so in this case, if we have this peptide bond forming, or amide bond, between two different amino acids, we would call this amino acid the N-terminal amino acid, and we would call this amino acid the C-terminal amino acid. And you would always add the next amino acid to the C-terminal. All right? So this one will always be the N-terminal amino acid. We'll talk about other instances where we can lose those amino acids. So now that we've talked about the monomers of amino acids, let's talk about how we can organize the actual protein molecules, right? The polymer of amino acids. So what are these levels of organization? So if we look in the graph over here, this is a good one to look at. Follow along while I'm talking. We have the primary structure just being the simple sequence of the amino acids that are connected by peptide bonds. So in this case, we have asparagine, glycine, phenylalanine, glutamate, glutamine, alanine, so on and so forth. That's the simplest portion. Okay. The next part is secondary structure. All right. Now this is just local 3D structure. So it's the arrangement of just a short sequence of the entire polypeptide into something that we're likely going to see as an alpha helix or a beta pleated sheet or turns and loops. All right? And this is important because it's stabilized by non-covalent hydrogen bonding. Okay, and that occurs between the peptide groups. Okay. The next portion, or the next higher order level of structure, we call tertiary structure. Now, tertiary structure is the overall 3D structure of the protein. So if we look here, we're now looking at this part. Okay, that's just a 3D structure. All right, and that's stabilized by side chain interactions between our secondary structures as well. And quaternary structure can only occur when you have more than one polypeptide. All right, so more than one polypeptide can come together, forming an overall 3D structure that is called the quaternary structure. So a little more in depth, our primary structure. That's our specific amino acid sequence, all right? We're looking back here again. Now it's connected by our peptide bonds or our amide bonds. And this is dictated by our mRNA codons. And if we remember back to the previous lesson, we know the molecular biology central dogma, right? So it's DNA to RNA. This is via transcription. And RNA can be made into protein via translation. So RNA gets turned into protein, so therefore the protein is dictated by the mRNA codons. And this is going to direct the subsequent organization. So the RNA is going to code a single codon for our cysteine amino acid, and then a serine, and then a leucine, and then a phenylalanine. So this is how it's dictating. Okay. Now, it's very important that even a slight change in the primary structure of a protein will affect its ability to function. And an example of this is found in sickle cell trait. All right? So the substitution of one amino acid for another in hemoglobin causes sickle cell disease. And all this is, it's a single difference in the 5th amino acid. It's changed from a proline to a valine. Now, a normal hemoglobin, okay, which is found in red blood cells, carries oxygen very well, and it has no effect on the shape of our red blood cells. So our red blood cells normally look like this. All right, they look like a nice little donut shape. Well, when we have this proline to valine substitution, that causes the amino acids, okay, to chain together in an unnatural way. Okay, so it causes the proteins to stretch and alter the color, or sorry, the shape of the red blood cell molecules. Well, when they're looking this sickle shape, okay, they can end up blocking blood vessels because they don't flow through nice and easy like a rounded donut-shaped one. So they can block small capillaries and veins and cause blockage, which can cause pain or possibly even death. So this can come from just a single substitution in a single protein, so a single amino acid substitution. Now, our levels of organization continue with secondary structure. And by the way, a way to describe structure is by going like this. So this would be primary structure. Secondary structure looks like this. Tertiary and quaternary. Okay, it's just that little degree sign. So our secondary structure I'll write it over here once again, primary. This one's actually secondary. Okay. So our secondary structure is just our local 3D structure. Okay, and it includes things such as our alpha helix or our beta pleated sheets and even our turns and loops. Now, our alpha helices and beta pleated sheets are held together specifically by hydrogen bonding. So if we look here, these yellow, it's hard to see on here, but these yellow portions are hydrogen bonds. Okay, these are all hydrogen bonds that are keeping this helix together. So what about proline in secondary structure? Well, proline is going to look like this. Okay, so this is what proline is going to look like. And as you can see, it's a very bulky secondary amine. Okay, and if we talk back here, right? So this is our alpha carbon. Okay, so we have our amine group. It's all attached in one, so it's not free to be working around. And what we have is that this is actually considered an amino acid, not an actual amino. But what this does, this actually is not able to be fit in a nice alpha helix. Because what it would do if we incorporate a proline, it would break that helix. Right? So what it's often found in is our beta pleated sheets. So our beta pleated sheets normally run like this, right? It has a nice little turn. Okay, and then maybe we go like this again, a nice little turn. Okay, and our prolines are often going to be found in the turns because they help with the U-turn portion of this. Right? So this is important to know, that proline is not going to be found in alpha helices. To continue on our levels of organization, our tertiary structure. This is our overall 3D structure of a protein. All right? And what's important is that 3D structure is related to function. Okay? Now, what that is saying is that structure dictates function. So if it looks a certain way, it's probably going to perform a certain action. All right? And we're going to talk more and more about structure dictating function as we go throughout. Now, tertiary structure is stabilized by hydrogen bonds, ionic bonds, hydrophobic interactions, and our disulfide bonds. And if we remember correctly, our disulfide bonds are found in our cysteine residues. Okay. Now, our quaternary structure only can occur when we have more than one polypeptide, so more than one subunit. All right, so we're looking at right here. The red, all right, the red is its own polypeptide. And then this other multicolored one is its own polypeptide. When they come together and form a complex, that now is a quaternary structure. Right? So hydrogen bonds, ionic bonds, and hydrophobic interactions all stabilize quaternary structure. So what will ultimately determine a protein's native structure? What do we think? Well, first of all, the protein's linear amino acid sequence is going to determine that. Okay. And what structure is that? What higher level order is that? Primary, tertiary, quaternary, secondary? Okay. Our linear amino acid sequence is primary structure. Okay. Now, what else will determine a native protein structure? Well, when we're talking about protein native structure, what are we even talking about? Tertiary structure, right? So this question is asking what determines our tertiary structure. And what is our tertiary structure? Right? That's the overall 3D, the total 3D depiction of it. Right? So that's tertiary. So the answer to what ultimately determines a protein's native structure is its linear amino acid sequence. So it's primary structure. And remember, structure dictates function. All right. Don't forget that one. We're going to come back to that multiple times. Okay, so proteins, once they're made, once they have their tertiary structure, they can fold in a whole bunch of things, right? So they can be globular, right? So they can look like a ball. They can be fibrous. Okay, so they can look like a filament or a string. There's a whole bunch of ways, and this is just a short, small sampling of what they can look like. But proteins can fold into many, many different shapes. And they're often helped folding by what are called chaperones. And these are other types of proteins. Okay? Now, if a protein is poorly folded, sometimes you can unfold it, which is called denaturing, and try to refold it again. That oftentimes doesn't help. So what you end up having to do is break down that protein and basically throw it away. So you degrade it by sending it to either the lysosome or the proteasome. And those are basically like that's like the cellular trash can. All right. Good. So when we've made our proteins, we can talk about what happens next. All right, and we're not going to talk about the reactions right now, but we can alter what happens to a protein after it leaves the ribosome. Okay, so that's when we're altering a protein after it's left being synthesized, right? So after translation. That's why it's called post-translational modification. So these post-translational modifications can regulate their protein's activity. It can regulate its localization, as well as how it interacts with other molecules. So it can alter its structure. It can alter its function. Right? And some common types of post-translational modifications are glycosylation, and that's the addition of a sugar. We can have phosphorylation, which is the addition of a phosphate group. We can have acetylation, that's an addition of an acetyl group. Methylation, an addition of a methyl group. Lipidation, okay, acetyl, methyl, okay. Lipidation, that's the addition of a lipid. Maybe we know that better as a type of fat. Okay. Ubiquitination is the addition of a protein, a small protein called ubiquitin. Ubiquitin. And then proteolysis is just cutting the protein. So maybe the protein, you have the protein amino acids, okay, and you cut off the first 5. All right, that's a way. Now, proteins can be classified in many different ways, and one of the easiest ways to classify them is based on their function. So you don't necessarily want to classify them based on what they look like, but you want to classify them based on what they do. All right, and as we can see here, a majority of our proteins are going to be ligand binding, DNA binding, or enzymatic. All right, and then the rest of them can be broken down into many, many other things. All right, so an enzyme is going to catalyze a reaction. DNA or RNA binding, self-explanatory, it's going to bind to our nucleic acids. Ligand binding and protein-protein interactions, it's going to bind to either another protein or maybe a compound in a cell. And then we have plenty of others that serve, say, structural purposes or make membrane proteins so things can go in and out. We have transcription regulators, so otherwise known as transcription factors. They can either increase or decrease the speed of making an RNA and therefore the making of a protein. All right, some are involved in contraction, transport. Some proteins are actually hormones, like insulin is a peptide-based hormone. We have storage. Okay, some are used as protection. All right, there are many, many different types, but the majority are of these 3 types. And since we're talking about enzymes, and we've talked about it before in Unit 1, enzymes are biological catalysts. So most metabolic processes require catalysts, right? So they need some form of catalysis. Now, our enzymes, if we remember back from the first unit, accelerate reactions, and they do that by decreasing the activation energy. Now, that activation energy is the energy required to get over the energy barrier. So that's what allows you to get to the transition state. Now, what's very important is that enzymes increase the velocity of reaction so highly that uncatalyzed reactions may never actually occur. So these can be increased on the 100,000-fold to 100 over trillion-fold. So this actually allows you a reasonable amount of time for a reaction to occur in the presence of an enzyme. Now, most biological catalysts or enzymes are proteins, but not all of them are proteins. Okay? Not all of them are proteins. Some enzymes can be what are called ribosomes, and that's where the active component is in RNA molecule. It's important to note that enzymes have a specificity of reactants, okay, meaning that an enzyme is only going to allow a certain thing, a certain compound to bind in it. Okay, so for example, if my hand is an enzyme, and the pen is a reactant. It fits in there just fine, and now the enzyme is free to catalyze the reaction, and the product of the reaction is writing on this slide. Now, if my hand enzyme were to pick up, let's say, my notebook as the reactant, do I get the product of writing on this slide? No. So the enzyme is specific to the reactant. It's only going to catalyze a reaction when it has the correct reactant in it. All right? And then finally, enzymes have an optimum pH and temperature, meaning they work at a certain environment. All right? So the cells in our body at a pH of around 7.4 have enzymes that are happy at about 7.4, right? And 98 degrees Fahrenheit. But if you were to throw those enzymes into our stomach acid, which is around pH anywhere from 1 to 4, those enzymes would not work very well. In fact, they would be denatured and then have to be degraded and thrown away. All right, so this is important. So for our final slide, we're going to talk about just a quick example. And I want you to arrange the following 3 properties from least similar to most similar between related proteins. So what I'm asking you is, between 2 proteins that are related, what is the most likely sequence, okay, that's going to be similar between any 2 proteins? Right. So let's say, remember, we're looking at the 3D structure of a protein, right? So the structure dictates function. Meaning, multiple things that look alike can do the same thing. So we might see then that the protein structure is going to be the most similar. All right? Now, if we remember back to the second unit, when we talked about the genetic code, right? We have DNA being made into RNA, and that RNA being made into protein. All right? Okay, and if we remember correctly, to make a protein, we need our amino acids. And if we remember, multiple amino acids, okay, multiple different codons of RNA on the RNA can be made into the same amino acid, which we call the degeneracy of the genetic code. So therefore, our amino acid sequence, we could get, let's say, aspartate from multiple different pieces of RNA triplets. Okay, so 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay, and this is RNA, our codons. And remember, our RNA is made from DNA. So this will have come from 1, 2, 3 of DNA, right? So on and so forth. So this should allow us to understand that what is next most similar between related proteins would be their amino acid sequence. And then therefore leaving us with the least similar being our nucleotide sequence. All right, so from least similar, we'll say similarity. So similarity increases as you go from left to right. All right, and we could add in another one over here in that what could be even more similar would be protein function. Would go right here. All right, so that ends our lesson for today. Thank you for watching educator.com. Please come back and see me again soon.